This is a production of Cornell University.
So it is my pleasure to introduce Hannah Zonnevyll, who is a second-year PhD student here in soil and crop sciences. Hannah got her bachelor's degree from the University of Michigan, where she worked on topics including moose herbivory in northern forests. Hannah then went on to live and work in Chicago for 2 years, where she worked for an environmental education nonprofit and also was a community science fellow with the American Geophysical Union. Here in SIPS, Hannah has served as the Graduate Student Council Chair, the Soil and Crop Sciences Graduate Student Assembly Vice President, and as a representative on the SIPS DEI Council. She also regularly leads outreach activities and finds way to create broad and inclusive science-focused communities. Today, Hannah will be telling us about her dissertation research, which is supported by an NSF Graduate Student Fellowship. Please join me in welcoming Hannah.
Thanks for that kind introduction, Dan. And hello, everyone. If I haven't met you yet, my name is Hannah, and I'm a PhD student with Dan. And today I'm super excited to share with you all the work that I've been doing since coming to Cornell, which is leveraging remote sensing to quantify pollen production across space and time for Juniperus ashei. Before I jump into the nitty-gritty of my project, I wanted to first give you a little bit of background about me. As Dan mentioned, I hail from the great state of Michigan, where I attended the University of Michigan and fell in love with plant ecology through a series of undergraduate experiences there. After graduating, I wanted to do some more community-focused work, and so I did an AmeriCorps service year in Chicago with an environmental education nonprofit that partners with Chicago public schools to bring a comprehensive, free of charge, outdoor STEM education to students from low-income households. And it was during this experience that I saw firsthand how the systemic racism in US leads to really awful inequities in terms of educational opportunities, access to nature, and ultimately public health outcomes. And so when I was thinking about returning to graduate school, I wanted to find a way to unite these two passions and ultimately do work that could help alleviate some of the public health burdens that I was witnessing. And that is what brought me to the Katz lab here at Cornell. And we focus on plant-related public health problems. And the one that I focus on specifically is that of airborne pollen. So who among us has pollen allergies? Raise your hand. Oh yeah, okay. Well, don't worry. Misery loves company and you are not alone. It is actually estimated that over 80 million people in the United States have seasonal allergies. And the worst that most of you will suffer are, a runny nose, maybe trouble sleeping for a few weeks out of the year. But for people with chronic respiratory issues, these asthma allergies and subsequent pollen exposures can be dangerous and result in emergency department visits due to asthma exacerbations. And we also know that asthma is not equally distributed across the United States. There's plenty of research that shows that asthma is in a higher concentrations within communities that are lower income. And so I have additional bad news, and that is that pollen exposure is increasing with climate change. We're seeing that the increased temperatures are leading to a longer growing season. What you're seeing here is the consecutive days above freezing, which allows plants to start releasing pollen earlier in the year and then continue later into the year. It's also resulting in range expansion for different allergenic species. This is something we're seeing in common ragweed in Europe. And a recent meta-analysis done in the Katz lab showed that plants are able to allocate more carbon to reproductive structures under increased carbon conditions. And so there's a variety of mechanisms that climate change is increasing pollen exposure and therefore possibly impacting downstream public health effects. Lucky for us, as with most things, science has the potential to offer a solution. And this solution can look like better airborne pollen forecasts. Airborne pollen forecasts can help people know when they might be exposed to pollen such that they can take their allergy medications with enough time prior to exposure so that it reaches its full efficacy by time that they are exposed to pollen. It can also let people know when to turn on their air filters or help them avoid pollen hotspots. However, current predictions of airborne pollen are insufficient to help people avoid this exposure. What you're seeing here is some work done by Dan that shows the predicted airborne pollen concentration from commercial forecasters compared to the empirically measured pollen concentration from pollen monitoring stations. And so pollen.com is doing all right, but the rest of them leave a little bit of room for improvement. And if you are associated with any of these companies, I'm not smack talking, I'm just inviting you to fund me. So with that, you might be asking, well, why is it that current airborne pollen is modeled so poorly? And one of the key reasons for this is because we just don't really know how much pollen is being produced across space and time. The current method for measuring airborne pollen is through a sparsely populated network of airborne pollen monitoring stations. There are only about 50 in the contiguous US. And so it does a really bad job of capturing the spatiotemporal variation in pollen production. And it is really difficult to calculate the amount of pollen being produced based on ecology of plants because it's super hard to study how plant reproduction varies over large spatial extents. As you can see, the historical way of doing this is manual measurements that take a very long time and actually can limit studies that involve counting these reproductive structures on plants to sample sizes of only 10 to hundreds of individuals. And further, it only covers a small portion of the tree and a small portion of the range of these plants. Lucky for us, we live in the time of remote sensing, which is an amazing tool in our toolbox that we can use to study ecological processes across the entire ecosystem. And there's been plenty of literature in the past couple of years that uses aerial images to track the timing of plant reproduction. And so it has been posited that we could perhaps use aerial images, such as with drones or with satellite images, to quantify the number of reproductive structures on plants such that we could calculate pollen production that we could use to inform these better airborne pollen models. So as you can probably guess, my research focuses on one central theme, which is building better airborne pollen models. I've just proved to you all that this could definitely help public health outcomes and improve the way that people interact with plants. But in order to do this, we need to have a better understanding of pollen production across space and time. And to help do this, I have designed 3 chapters to pursue here at Cornell. The first one is to actually measure the amount of pollen production over space and time using remote sensing. The next is to build off of the data collected in chapter 1 to then start to disentangle the abiotic drivers of plant reproduction. This is possible because the chapter 1 data set will let us quantify the number of reproductive structures on plants that we can pair with gridded environmental data and start to model what might be the dominant drivers of plant reproduction. And in my third chapter, I hope to take a ecological forecasting approach and actually try to predict how climate change might affect future pollen production of these plants. This is a similar approach that has been done in phenology, but has not yet been done for pollen production. I'm going to spend most of my time today talking about chapter 1, since that is what I have worked the most on so far. But I will also give you a little teaser into chapter 2 towards the end. With that, let's get into it. So I'd now like to introduce you to my study species, the beautiful ash juniper, or Juniperus ashei. Juniperus ashei is an evergreen dioecious plant that is the dominant tree species in central Texas, where it thrives on the calcareous soil of the Edwards Plateau. And It is known by locals as an infamous producer of pollen that causes something known as cedar fever. And cedar fever is an awful affliction that results in severe runny noses, achy joints, trouble sleeping, and it can actually even cause some people to run a fever. And the culprit of cedar fever is the super abundant number of reproductive structures on these plants, which are pollen cones. Male individuals can actually contain up to 1.3 million pollen cones on a single tree, and each of these cones can have almost 500,000 grains of pollen. And so this pollen is so abundant and so allergenic that past work in the system has revealed that it can contribute up to 18% of asthma-related emergency department visits during peak pollen season. So yeah, you can see the pollen coming off of these trees in waves, and it is, from personal experience, very unpleasant. So the thing that makes ash juniper a horrible neighbor to all of the Texans actually makes it a wonderful study species for me, because the pollen cones have very distinct visual properties. As you can probably tell, this individual, which is a male with pollen cones, looks quite different from this individual, which is a female having no pollen cones. So let's take a little bit of a closer look at these cones. They have this kind of beautiful orange color that makes them stand out against the green foliage that you can see here. And so it is this aspect of the ash juniper that makes it observable from aerial imagery. In this case, I have been trying to derive methods that use aerial images from drones as well as manual methods to quantify the number of cones on these trees using their spectral properties. And I'd now like to show you a little bit more about what those methods look like. So to test my methodology, I have established 14 study sites across the range of the ash juniper trees in central Texas. You can see that the study sites cover a range of environmental conditions, including temperature, precipitation, and what is displayed here is soil moisture data from SMAP. And at each site, I choose between 5 to 10 focal trees on which I take a comprehensive suite of measurements. And I take additional phenology measurements from surrounding trees to try to understand how the timing of reproduction varies among these trees and how that might impact the spectral reading from the images. I have done this for 2 field seasons now, which I go to Texas in January. So it's a nice little escape from Ithaca winter, but unfortunately, that seems to last until mid-April anyway. So I was also very lucky this past field season to be joined by a wonderful UT grad, Dr. Annalisa Shields Estrada, who was not only a really hard worker, but an absolute joy to be around. So I'm really grateful for her help on this project. And so to show you a little bit more about the methods in practice, I first select my focal trees at each study site and take a couple of manual measurements. This year, this included the diameter at breast height and the height. I then put a quadrat with a known area on a branch of the tree so that it's facing upward and visible by the drone. And after I fly the drone, I harvest all of the foliage that falls within that known area of the quadrat. Then I use a super sophisticated method for homogenizing it where I run it through a mulcher, and that enables me to have a representative sample where I can subsample the foliage 10 times, take its weight, and count. And this will give me the number of grams of foliage, number of cones per grams of foliage that I can then multiply to the number of cones per grams of foliage for the entire tree by using allometric equations based on diameter, breast height, and height to give me the total gram of foliage for the tree. And so these are all my manual methods, but we're not done yet because we now get to do some fun remote sensing things. So from the drone images, I find the quadrats, prop that down so that it only contains pixel values that fall within that known area. I then extract all of the pixel values from that image and correlate these pixel values, their red, green, and blue values, to the known quantity of cones that I just estimated using my manual methods. This results in an index that can be used to predict the number of cones per grams based solely on the red, green, and blue values. So having this index allows us to extend our measurements of these reproductive structures beyond just the focal trees. And I'd now like to show you the index. So this is what I am provisionally calling the orangeness index. If you have any other recommendations, let me know. But it is a combination of the red and green values, red minus green over red plus green. And as you can see here, on the y-axis, we have cone density. And so this is the manually estimated cones that I used with my subsampling. And higher cone density results in a higher index value because there are more cones in the picture, more red values, more positive value. Compared to this quadra over here that has very few cones and a very low cone density, has a lot more green values associated and therefore returns a lower index value. So awesome, we have our index. What does it look like when you actually apply it to images? What you're seeing here is a quadrat picture that I have processed to only retain the pixel values that are either foliage or cone. I use a random forest pixel classifier. You can see the results down here, but it performs very well. And so when you apply the index to this, you can see that the positive values do indeed match up with where the cones are. For visualization purposes, I just created a binary. So the positive values associated with cones are orange, and the green values associated with foliage are negative and green. And so, yeah, you can see that it works pretty well and does a good job of picking up the distinct properties of these cones. But remember that the overall goal is to use this index to count cones from trees that we don't actually have the manual measurements on. So let's try to do that. Here you are seeing a subset of an orthomosaic image from the drone, which is basically just a stitched together landscape level picture that I put together all of the pictures from a drone at a study site. And you can see the varying densities of cones on these trees. So there's some with a lot higher density compared to some here with lower density. And what I've done is manually segmented these treetops so that we can only retain pixel values that fall within the canopy. And once again, I can extract the pixel values here and run it through the pixel classifier, apply the index, and here we are. So again, the lower values are associated with more foliage and the higher values are associated with a higher cone density. From here, we have all of the ingredients that we need to actually go ahead and quantify the number of reproductive structures and therefore the pollen production of these trees, because we can, again, calculate the total grams of foliage for these trees using allometric equations, where the gram of foliage is a function of the diameter, breast height, and height. And then we can take the average pixel value or the average index value from these trees, multiply it by the grams of foliage from the allometric equations, and this can give us the number of cones per tree. Using this, we can calculate the pollen produced, and this can all go back into creating better airborne pollen forecasts based on a more comprehensive measure of the amount of pollen being produced. So, well, I am definitely very excited about these methods and my work so far. There's plenty of limitations and it's not perfect. Some limitations that have been difficult to work through are that the trees have a kind of a wide variety of coloring in their foliage. So as you can see here, this is a tree that has a lot more yellow foliage rather than green. And so it's returning a really high index value despite there not being very many cones in it. Further, the index is sensitive to lighting conditions. So when we have pictures that are either very overexposed or very shadowy, the index is not able to do a very good job of calculating the number or predicting the number of cones based just on the spectral properties. Additionally, it has been difficult to calibrate the index using coarser resolution images from the drone, and so I'm still working out some kinks there. But when I'm thinking about next steps, there's a lot that I am looking forward to working on. So first, I plan to create allometric equations that are specifically using remote sensing variables. This is what these trees look like. So it is really not that fun to take the diameter at breast height of these trees, and also probably not that useful because it is not a variable that's observable from aerial images. So I plan to use LiDAR in the coming field season to create better allometric equations that rely on variables such as height and canopy diameter instead of diameter at breast height. And that will save me a lot of time and a lot of being stabbed in the eye. So I'm looking forward to that. And next, these trees, as I think with all other trees, are 3D objects. So they contain a lot of information on the sides of the trees that are missed from aerial images. So to account for the number of cones that are hidden from aerial images, I've taken some orbital flights around my focal trees, and I plan to create a conversion factor for the number of cones that are missed when just looking at the trees from the top down. Next, and this is kind of like my big push right now, is to figure out ways to adjust for different lighting conditions. Again, you're seeing here a quadrat picture that is just in a lot of shadows, and the index doesn't really know what's going on with the number of cones that it is seeing here. So if you have any kind of workflow that you've used in your research, I'd be very happy to hear about it. And hopefully I can find a workaround for this. And lastly, one next step that is feeling especially exciting to me is scaling up to satellite images using the index to be able to measure cone production and pollen production across much larger spatial and temporal extents. And this would allow us to get a more comprehensive data set across Central Texas at the regional level, rather than just being limited to the study sites that I'm able to visit. So I want to now bring us back to where we started. So I've shown you so far how we can better measure pollen production across large spatial and temporal extents. And so now that we can measure it, how can we use this information and pair it with existing environmental data to start to understand what the abiotic drivers of reproduction might be that drive variation in reproduction across space and time. So this is very new data that is more of a proof of concept than anything. But what you're seeing here is the mean index value from my sites compared with the mean soil moisture data from SMAP. And it is this kind of workflow that I hope to use in my second chapter to compare the reproductive output with gridded environmental data sets. Obviously, reproduction is an incredibly complex process that will require much more sophisticated statistical tools, such as hierarchical modeling, to start to uncover what is driving the variability in reproduction in these trees. But yeah, I'm excited to explore this in my coming years at Cornell. So key takeaways. If you remember nothing else from this presentation, my name is Hannah, and also I have just shown you that we can measure pollen production over these large spatial extents using aerial images, which will allow us to create better airborne pollen forecasts. That is part of my chapter 1. And then we can also use the data set collected here to start to explore the abiotic drivers of the variability in reproduction for these trees. And this will be part of my chapter 2. And you will just have to stay tuned for chapter 3. With that, I'd like to thank everyone who made this work possible. Everything we're doing is in the presence of others, and I'm so grateful for everyone, not only the people listed on this slide, but just to be in community with you all has been a real joy at Cornell. So thank you. And I'll take questions now or later. Okay.
I'm just curious, how did you arrive at your sort of orangeness index? And did you try other sort of indices?
Oh, yeah. Great question. Yeah, I tried a lot of indices, actually. It was a lot of like looking through the literature and seeing what other people have used. And we arrived at the orangeness index just kind of by way of it being what seems the most feasible, like because the cones are so orange, it's we thought that maybe because there would be more red values associated with those cones compared to the green values, that would be like the best combination of bands to actually pick up on those differences. But yeah, definitely we still could possibly change, but that's what's worked the best so far.
I was wondering, is there hope to, I mean, maybe the answer will come when you have more information on chapter 2, but is there hope to kind of do a survey 1 year and then run with that information for several years, or you think that it's going to be something that has to be done like yearly?
That's a good question. So like how often for data collection? I plan to do 3 years, but the idea is that if we could scale up to satellite images, this would enable me to not actually go down and collect the data, but have a longer time series, both, you know, back into before I started at Cornell and then when I don't have to go down anymore, and kind of like using that information as well. But yeah. Awesome. Okay. Thank you all so much.
So thanks again, Hannah, for a wonderful talk. And I'd like to introduce Dr. Dave Miller next. Dave is a postdoc working with me here in soil and crop sciences. And he did his undergrad at Boston University, and then he got a master's and PhD from UC Santa Barbara. He then went on to do a postdoc at UC Berkeley, and some of the topics of the work for his dissertation and for his first postdoc were about trees and cities and using remote sensing to understand the effects of drought on them. His work from these was published in journals including Nature Communications and Remote Sensing of Environment. And we've been very fortunate to lure Dave away from sunny California. And he has been instrumental in this transdisciplinary collaboration, the Cool Trees team, which is working to understand how we can use trees to keep cities cool during heat waves and ultimately reduce heat-related morbidity. And we've been very fortunate to have Dave here and to benefit from his expertise in remote sensing and in making beautiful maps. So please join me in welcoming Dave.
Thank you, Dan, for that kind introduction, and welcome everyone to our second of 2 talks today. It's a tough act to follow, and I'll do my best. So today I'm going to be talking a lot about New York City and classifying urban tree species using a lot of high-resolution satellite time series for heat-related public health impacts. A lot of our co-authors here are at Cornell in civil engineering and also at Weill Cornell in New York, and we'll kind of get into it. So tree species vary in cooling capacity, but how can we use this to actually reduce heat? Anyone who's walked under different types of trees in your neighborhood, a big maple tree is going to provide a lot more shade, a lot more cooling than perhaps a smaller cherry tree would. And so tree species actually cool differently in the environment, but these differences aren't really included in models for heat and human exposure health related. So shading, for example, tree size, of course, and leaf area, so how many leaves do you have on this particular tree, as well as evaporative cooling. So this has to do with transpiration rates, how much water can these trees actually pump into the air to cool things down over time, model density, things like that. So there's differences across species. But these aren't really included in tree selection decisions. The most you'll probably get from these sort of things is like, how big is this tree going to get? Is it going to get in the way of power lines or something like that? And also, is this tree actually going to survive in this location? But in terms of a lot of these species level differences, we don't really have that information. And we don't really have good information about what trees are where in the city of New York. We have some information, quite a bit, but we don't have information about where they all are, which is really needed to reduce heat and health impacts more precisely. So maps of tree taxa are really needed to reduce heat more precisely in cities. And because of this, we can actually estimate cooling at a scale that's more relevant to urban residents. If you're thinking about a large neighborhood area, a lot of this health analysis is done at like census blocks or census block groups. It may be shady and cool in one part of the neighborhood, but not the other part of the neighborhood. And that's an issue because if you're walking down the street in your particular part of town, if you don't have many trees in your street, what does it matter that that park has tons of trees just down the road? And so there are many methods from remote sensing to do tree identification, but getting down to genus and species level stuff is just pretty hard in general. Big issue is that midsummer, you may have experienced this yourself, green trees kind of all look the same, especially from multispectral images, like a lot of the satellite data we actually use. But at certain times of year, trees can look very different. So I'd like to show you one of the few windows in Bradfield Hall. This is looking north. Dan took a bunch of these pictures, and what you can see here is that these are a few different times of year. So summer on the top left, trees all look pretty green, but spring, early fall, late fall, they look kind of different. So if you're looking at this particular tree over here, well, it's turning yellow while other trees are senescing a little bit. But this one over here, it's not quite the same color as the other one. So perhaps they're different species. Maybe we can lean on this type of information to get at different types of seasonality. So can we use deciduous phenology to classify trees? And by phenology, I mean the seasonal timing of different events in nature. So in the case of deciduous trees, when do they put out leaves? When do they lose leaves? How does this timing differ year to year and between species? So urban trees have been classified from multiple seasonal images recently. So you take a big stack of satellite data, you throw that all into a big classifier. You can do pretty well with that. But what there haven't been before is that classifications based on the time series that actually derives the phenological dates. So like what is a precise date you could give to a model to say, oh, what is the leaf out in the start of spring? So what we can do is we're aiming to identify taxonomic differences at fine timescales, which all sounds well and good, but actually getting the data to do this is not the most straightforward thing in the world. So there are kind of some data requirements to this. One of the big trade-offs a lot of the time with satellite imagery is different types of resolutions. Usually you can get pretty good temporal resolution, so lots of observations, but the pixels have to be pretty big. But if you're dealing with something that you want to get lots of precise measurements, you need high spatial resolution as well. So fortunately, Planet is a commercial satellite company, and they have a constellation of satellites called PlanetScope that both has high spatial, roughly 3 meters, and high temporal resolution imagery. So you can get observations almost every single day. And the way they get around this is by launching hundreds of these small sats that take observations pretty often. So we can do that and lean on that to look at individual trees and their seasonal timing. But we also need good labeled training data to actually predict on. You can't do a classification without having some good training data. And thankfully, the city of New York has tree stem and ID database from a street tree database where people literally went out and hugged 1 million trees in New York City and labeled them with different genus levels, cultivar, quality status of the canopy, which is awesome, but that's still less than 20% of the total number of trees in New York. So there's still a lot of places that need to be filled in. And also you need a good tree crown polygon data set because you actually need an object that you're putting a label on. Otherwise you're just doing pixels and it could be pretty noisy. So thankfully, there's already been classification of tree crown polygons from citywide LiDAR flights. We tried to make polygons ourselves from LiDAR. It's pretty challenging, and so we're thankful that this data set already exists. So the research goals from this, examining the differences, the first step in this, and examining the differences in urban tree phenology by taxa in New York City. And so the main research questions for this is how can satellite-based phenology be used to actually separate urban deciduous tree taxa in New York City? One of the key things to keep in mind and why we're getting away with this technique in a place like New York is that it's mostly deciduous trees, like over 90%. If you were doing this in a place with a lot more evergreen trees, like let's say Seattle or Miami, you'd have a lot harder time doing this, and this may not be the best technique for you. Some other questions, temporal. So what is the year-to-year variability of spring leaf out, like in terms of the start of season among different types of trees? And also spatial. So how does spring leaf out vary spatially across the city? Maybe you're a tree in Manhattan, you're a real go-getter, you leaf out early. Trees in Brooklyn, maybe not. I don't know. So we'll have to just take a look at it. See. We're also going to look at the end of season, but it's going to be a lot more challenging. So here's an example of tree data in Flatbush, Brooklyn. This is a little bit south of Prospect Park. What's cool about this neighborhood is that you have a lot of single-family homes with trees individually across this sort of central neighborhood, but larger apartment blocks over on the side. So big differences in the amount of tree cover you have. Each one of these colored points is a street tree that the Parks and Rec Department actually labeled, and so different colors are by different tree species. So There's a lot of these points, which is awesome, but there's still a lot of places that are missing. So this is what this tree polygon data set looks like on top of that. And this is from the Nature Conservancy and the University of Vermont Spatial Analysis Lab. So this data set is really cool because we can actually put labels to all these trees for the places that are missing. So how do we actually get there? We have this map of tree stems from the New York City Parks and Rec, and what we want is a final map of all the trees labeled in New York City, right? No big deal, it's fine. How do we actually get there? We use this PlanetScope satellite time series, LiDAR tree crown polygons and structure, and also air temperature data to look at what time of year should trees leaf out based on accumulated, how warm is this different place in the city. So for each tree crown polygon, extract this time series of reflectance from PlanetScope reflectance time series, clean this up, extract key phenology data, and then also LiDAR structural variables, so like how tall is the tree, how big is the crown, how dense is the canopy, and air temperature related data. And so we have this suite of different information all for this individual tree. So we can use the labels of tree stems from the New York City Parks and Rec as our reference training and validation data set. So this is the trees we know what they are and where they are. And we can use that and develop a classifier. Yes, I'm using random forest to classify trees and apply that classifier to all the unknown tree polygons across New York. And ta-da, we have a New York City training data set. All completed for a final map of trees in New York, but I'm going to talk mostly about this part up here in terms of actually how we get the phenology information out of this. So here's from the National Agricultural Imagery Program, NAEP. If you've looked at any agricultural data, you may be familiar with it. They do it in New York City as well. So this is a true color air photo from summer 2019, which is great, but they only do this every couple of years and right in the middle of summer, so you can't get much seasonality out of this. So this is PlanetScope, which is slightly coarser resolution, but much more observations. I'm showing it in a false color composite, near infrared red, green, with near infrared on the red display, so plants reflect way more light in the near infrared. And so anything that you see on this map is bright red. That's normally green plants that you would see with your naked eye. So here's phenology time series of a particular oak tree, a very charismatic one that's well behaved in the South Bronx. So that's what it is in the NAEP imagery on the top left from the street. And so this particular tree, I'm showing the PlanetScope imagery down here. And this is a time series of NDVI through time. And you can see as it gets bright red in the spring, that's when it's greening up. And so this is what we're tracking for every single one of these tree crown polygons across New York. And the actual value we're tracking is the NDVI, which is a very common satellite remote sensing index where you're comparing the near infrared to the red. As this number gets higher, the plants are greener, and we can track this year over year. So for example, for all the trees that we know what they're labeled, this is for all the maples in New York City for 2018 from PlanetScope. So this, you can see a lot of these are towards the first week of May, roughly, but you can do this year over year. And so you have approximate timing of leaf out variability across every single year that we have available. You'll notice there's some year-to-year variability. For example, 2019 and 2022 tend to be a little bit later. But what we can see is that when we can separate this out further, you can get down to species level as well. So for example, silver maple in the middle here tends to be a little bit later than red maple or Norway maple. So there are some different species level differences we can get at as well. So we can do this for other genera. So for example, here is platanus. So they're plane trees or sycamores. One thing you'll notice about this one is it tends to be a lot later than the maples, which is reassuring because they probably should be. But also this year, 2019 is super duper late. So we're looking at like late May for this tree, which is suspiciously late in some ways. So how do we actually translate phenology into a classification? One thing to keep in mind is that this is one particular year in 2019, and this is just separated by different genera, and this is different months in the spring, box plots showing the distribution of timing for the trees that we know. So for example, based on this, we could do a pretty good job separating Glitteria, the honey locust, from Acer or maples. But getting back to platanus, it's way, way later than everything else. So for example, we should be able to readily separate this genus based on its consistent late spring leaf out in 2019. What we think is going on is that there is an anthracnose fungus causing delayed spring leaf out. This is a fungus that causes the plants to attempt to leaf out in the spring. Then it looks like they have frost damage or leaf wilt, and the leaves kind of shrivel up and don't look that happy. Trees look bare until much later in the growing season. Then they get their act together by the end of May, and summer goes on. But what we can do is that for an example test case on a classification, we can lean on this and a few other structural variables from the LiDAR and do like a prototype classification for the Bronx for platinus versus other genera, just a split. And so we get like a 93% overall accuracy in this. The big points that you see on here are the actual labeled street tree stems. And so everything else, we label it as either platinus or something else. And so the blue, you can see a lot of platinus are labeled in a lot of buildings where around buildings where we don't have the street tree labels. So we're painting everything in that we needed new labels for. And we can do this in a lot of bigger neighborhoods. So for example, this is a relatively large apartment blocks. And you can see that a lot of the trees that weren't included in that street tree database, we can actually provide labels for based on this. So we can do an initial test case on this, but what about spatial variation in phenology? So we need some more information based on temperature as well. So this is a temperature data set that takes coarser resolution data, downscales it based on different structural parameters on the surface, like green vegetation, impervious surface, things like that. We didn't make this product, but what's really cool about it is that it's really high spatial resolution, so we can get pretty good information on what is the temperature accumulation for different trees. So what's important about that is that if you have different amount of temperatures in different locations in the city, you would expect, oh, if I'm in this location and it's this warm, well, what would my leaf out timing be for this genus versus this genus? If we have information about, okay, it leafed out here and it's this warm, well, we have a much more better information about which is which. So for two different genera, we might see different response patterns in air temperature. And this is where we're kind of going next steps for this. One other thing that I've been dealing with the last week or so is looking at a webcam. This is what's called a phenocam, so phenology camera. This is at the top of CUNY in northern Manhattan. And this is just staring at this park all the time. So this is St. Nicholas Park. Here it is in, this is beginning of April 2022. What they look at is like, they take a polygon like this and then work through it through time. You can see the trees green up. And so just back of the envelope, comparing this sort of polygon shape that they have versus the trees in this polygon, 2022, we're roughly the same time, May 3rd, April 29th, which isn't too bad. 2023, we're a couple weeks off, but in digging into the data, there may be some issues with the phenocam, and there may be some issues with the PlanetScope imagery, so take that one with a grain of salt for now. But what I'm working on now is examining individual trees, so taking little boxes of trees and tracking those through time individually. So just some implications for this. Phenology shows promise as a way of identifying urban tree taxa. So we have this platinus classification as a proof of concept, and there's separability that currently exists for other genera, but we're going to build on this and include temperature phenology relationships and lots of other things in order to do a better classification. And so the goal will be, well, we're not going to get every single species across the entire city. There's just too many of them, but we can get a lot of the most populous species. We won't get populous, that's not in here, but we'll get a lot of other things. So methods have potential to be scaled spatially and temporally. So we could do this across multiple years if we needed to. And the eventual goal will be to create a scalable pipeline that can be applied to other cities. So if you live in Cleveland or Chicago or, I don't know, Ithaca, if you had the data, we could do something like this, which would be pretty cool. You'll notice I didn't talk about health at all for the last, I don't know, 16 minutes. So what's the big picture for this? We're going to link tree identity to public health decisions. And so my corner, Dan's corner of the project is in this tree taxa classification, mapping trees in New York using the remote sensing and the phenology product. But what we're actually planning to do is hand off this information to colleagues in civil engineering and Professor Chi Lee's group to do genus level classification. Take that genus level classification and turn it into an urban heat model. So they have a model that can run at pretty high spatial resolution, roughly 100 meters or however much they want to define it, and include that information on tree structure and cooling to be able to have better information on what is the condition in different neighborhoods at a scale that's relevant to people. So when we have that sort of temperature and heat information, that can be used related to epidemiology and social justice. So assessing marginal effects of species on like emergency department visits, like if you have certain amount of certain types of trees in a neighborhood, comparing those neighborhoods, how many department visits do you have during heat waves or other times of year, and also those different types of health effects of these trees into urban greening decisions and public health policies. So that includes colleagues at Weill Cornell, including Arnav Ghosh and Ashei, colleagues in natural resources, and also in other parts of around New England as well. And so additional acknowledgements, colleagues at the Nature Conservancy and the Natural Areas Conservancy, two separate organizations with similar sounding names, and some other information were from colleagues at American University and CUC Santa Barbara to help with the plain scope data. So I'm gonna leave this up for the end, and feel free to ask any questions, and thanks for your time.
So many cities are considering what's going to happen with climate change and how that's going to have to change the kinds of trees they plant in a given location. Are you contemplating those issues in your work?
Yeah, so the question is about, with climate change, is that information in terms of what the tree species palette should be included in our work and how we're looking at that. We're not addressing that specifically, but that is definitely a question longer term. And it could be something where you look at different parts of the city and say, oh, this part of the city is warmer. You could use the survivability in this part of the city to compare and project in other parts. There's stuff being done in terms of like moving different genotypes and planting those that are from further south. So we're not dealing with that end specifically, but that's definitely a consideration for sure.
I really like the animation, the time series animation with the DVI going up and down. I was expecting that thing to kind of plateau and stay kind of high, fairly level, but it's kind of, it peaks and then starts going down.
Mm-hmm.
Is this typical of all trees doing this, or is this something that could be used for maybe differentiating trees?
Yeah, so that's pretty typical for trees.
It peaks and then it's
Yeah, and you get a peak in like around June-ish, and then it slowly kind of declines through the summer. It's still a dense canopy through the summer. It's just they're not as bright, vibrant as you would have first thing in the growing season. So yeah, that plateau isn't necessarily totally flat. Yeah.
really cool.
That was great. Thank you. It's so nice to hear more about what you've been working on. Kind of related to this last question, I'm curious if like there are other like land surface phenology metrics besides just start of season and end of season that kind of describe that curve in more detail. And have you guys looked at how some of those other metrics might vary across the species in terms of helping to parse those out?
So the question is about other land surface phenology metrics besides just start of season and end of season. We've looked into it, but we haven't actually gotten too far with that yet. So there's other stuff we can fit in that curve besides what I'm showing as start of season, end of season. It's just like the 50% point on these S-shape, like these sigmoid curves. But you can do different times a year, and a lot of these other phenology models will do stuff where they have like, here's the max, here's this mid slope decline, lots of other things like that. I think for now, we're just going to stick with these and then build out from it. One thing we actually want to try to do is actually lean on the color component of it more and see if we can pick out individuals that have really distinct color situations in different parts of the year. For example, like ginkgo trees, like that ginkgo tree out there turn like this electric yellow in the fall. And if we can capture that, that would be really helpful. So yeah, I want to think about that more for sure.
Does your system allow you to capture like invasive trees? I mean, I'm thinking, New York City, tree of heaven, Bradford pears, which, a lot of folks certainly wouldn't want to see, or at least an increasing number. So I'm just wondering, have you come across that as being an issue?
Yeah, I mean, so the question is about, can we capture invasive trees? And the answer is if they're in the data set already and there's enough of them, we can probably label them. We don't have 100% like what the classification level is going to be, but tree of heaven, Atlantis, there's enough of them that we should be able to get those. But if they don't currently exist, so there's very few, they won't show up in the classifier. Yeah, unfortunately.
Nice talk, Dave. I have a question. Coming back to your very original motivation, like the different cooling effect from different species.
Mm-hmm.
I was wondering how your classified map can be combined with ET, like Yun is doing, to do the optimize like spatial distribution of different tree species to basically to best mitigate the heat wave in the future. I think that's probably most important. And also the second question is, have you considered the air quality impact? Because a lot different trees emit like isoprenes, right? That's a huge air quality issue. So I think the optimization, like with the different cooling effect and also air quality, should be included in your like the downstream work.
Thanks. Yeah. So the first question is about looking at different species types and evapotranspiration and how their cooling will be included in the model. We're talking about that, and we're not 100% sure how that's going to slot in exactly. The first factor is leaf area index, of course, which is just the amount of leaves you have over the surface. But what we would like to do is look at like the TRI database and look at different species cooling capacities and be able to work with Professor Lee to include what we can in the model. So that's still a work in progress. The second question is about air quality measurements. And I know Dan's very interested in air quality. So yeah, we've been talking about some of the stuff related to pollen and but other things related to like VOCs. We'll talk about that more because there's a lot of cool implications that can come out of a classification once we have it. But yeah, thanks.
All right.
This has been a production of Cornell University, on the web at cornell.edu.